Right. So again, the most important thing in CellPathfinder is that whenever you start, you have to select the parent folder. Now, you can go up to, I think, about 2 parent levels ahead, so it could select a different folder, and that will open up additional data. But in this case, I just want to limit that. So normally what you do is you'd have a folder like on your server, and it'd be like the CQ1 folder. And then in that folder, you'd have a bunch of different users, and each user or each series of experiments would have a different subfolder. So then you would just select like that subfolder, and then you'll be able to open up all the data sets in there. Does that make sense?
Yep.
Everybody's like, oh, okay, I get it. Kind of follows. Sure. Yeah, and then whenever you want to open up in particular data set, now one of the first things you'll have to do, I don't think I have any. Let me see. Let me just do a quick check. I think all of these data sets. These have already been converted. A lot of times, whenever you start on a data set for the very first time, you'll see that it's there, but then if you click on it, nothing will appear. And so what you need to do is you need to either use the convert or the align function. And so the convert basically double checks the data's there, and it writes the metadata in a format that CellPathfinder can open. If you have tile data, so you've got multiple XY positions, you need to use the align function, which does both convert and alignment. So if I go to here on the tile data set and I hit align, now it's going to come up and say skip because the data set has already been aligned. That's kind of important whenever you have kind of larger structures. So if you're looking at large organoids with multiple fields of view, and you need to stitch those together, you can use the align function to tile them and treat that as one data set. Does that make sense? Are you guys do you remember being trained on the tiling function?
Mm.
Yes, no, what's that? Yeah.
I think it's just a Z stack, or
no, not a Z stack. So whenever you so actually I'll open up this data set here, this tiling. So by default, if you look at a particular file, you'll see, oh, here's just a single field of view. But you can do tiling. And so if you use the alignment function and we open up the tile, you can see that this is not just a single XY position. It is a multi-position thing, which actually brings us to the second most important thing. And the one guy is missing. So we're going to do a quiz later, and I'm going to ask you what the second most important thing is. And because that other guy behind the NASA MacBook is missing, we're going to quiz and we're going to ask him what it is. So, so the second most important thing, again, is over here in the right-hand corner. And this is this button right here, which is the view setting. And whenever you toggle that, you can get access to the contrast adjustment. This is where you change the brightness and the contrast for each channel individually. And so you can go in there and you can adjust. Is everyone familiar with brightness and contrast? Yes, no?
Yep.
Yeah, okay. It doesn't change the data. All it's doing is changing how the data appears on the screen. So it makes no difference to the actual data, makes no difference to any of your segmentation or analysis. Doesn't change anything, but it makes it easier to see. And you can turn it off or on. So, okay. So anyway, so that was the second most important thing. So again, so what can everyone tell me? What's the most important thing? Who's the person with the MacBook and the NASA thing? Yeah, you. So do you remember what's the most important thing?
Trust the parent, not the child.
Yes, good. Okay, that's 50%. Now, what's the second most important thing?
I don't know. Find a little
brightness contrast.
Yeah, brightness.
I just had to leave the room. She has it.
Yeah. Okay, so in the so whenever we open up a data set, so I'm going to go open up. I'm just going to pick on the cell painting data set here. So whenever we open that up, you'll see over here, by default, it opens up like this. But this little button right here, I don't know if I can make my screen. I can't really make my screen any bigger. But there's this little button. It looks like a view setting with a region and a contrast. It's a little green circle and some blue bars, that's the second most important function in the software, because I find I use this all the time. And I come in and open that up, and that brings up the the contrast enhancement and the region settings. And so from here, you can go in and turn off the different channels. So I consider this kind of the second most important thing. If you can get these two things, which are poorly labeled and hard to find, everything else in CellPathfinder is like as easy as eating donuts, pretty much. Yeah. So that's kind of, yeah. So this is pretty important. The other thing I like to tell people is to watch out for automatic, because automatic will frequently hone in on maybe like the wrong thing. So you have to be very careful. Sometimes what you think is good is not really good. You have to actually look at the real numbers in the data. So you just have to be really, really aware of what's going on. You know, because sometimes you'll get things that look really good or bad. I should have a canned bad data set. I feel like a lot of times somebody has a data set with some hair in it, and hair was like super fluorescent for whatever reason. There's a lot of like hair that gets in these multi-well plates, and then they're like, why can't I see anything? It's because there's like a piece of hair in there, and it totally throws off the automatic contrast settings. But anyway, that's neither here nor there. Okay. Getting back to it. So I want to pick up this data set, dots and cells, and just kind of go over the basic functions of the software. And so we basically start at the top, gone to the next row, talked about left to right. So now I'm going to open up data set. I'm just going to open it up. And then what we're going to do is we're going to start. We can either start I normally start in view. So we're going to talk about some of the different functions, where stuff is, and then we're going to go back and talk about the protocol, which is basically the business end of things, where you set up your image analysis protocol. You basically build your segmentation protocol, which identifies the different parts of the cell, makes your measurements, and gives you your results. So we're going to do the view first. So we have things like viewing, thumbnail image 3D, cell information, cell charts. Now the cell charts, all the charting functions are only available after you've done the protocol, which is why, open the data, do the protocol, set the view. That's why view is next. But I like to start off with view to familiarize people with the interface before we go back and talk about the image analysis protocol. Okay, so again, we're going to go over the second most important thing, which is over here, which is the view settings for the region and contrast. We can turn on and off the channels. If you had objects, you could turn them on and off here. We also, so that's kind of the second most important thing right there. I'll turn that off for the moment. Again, starting from, so we've gone down here. If you had multiple wells, you can use, we've got a bunch of different places that you can change the well. Like if you don't find one place, there's like at least 2 or 3 more places you can change the well or the field. So the first one is up here. So if you have time series, you can use T is for time, W is for well, F is for field. We can select the different fields. If you have multiple Z-planes, you can change the Z-planes here. So this is the first place. This one is the autoplay. So if you want to go through and play like different time points or something, I guess you can. No, you can't play different wells. It only plays time. Oh, you can play the wells. So I can click on well here, and it'll go through, and it's supposed to swap between the different wells. I mean, if you want that, I don't know. It's a bit silly, but it's there. Okay. There's also the option to save images and save movies from the report. We'll come back to this later. You can also adjust the contrast here. However, if you make any changes, it kind of gets hidden immediately. I don't like using this because, I mean, it's nice if you want to make a quick change, but if you want to go back and adjust it, it disappears. That's why I always use this one, because that's persistent. Okay, so in the navigation, so we've got navigation and properties. We'll come back to properties after we've done some segmentation. But here we can see if you want to load protocols, we can load the protocols there. We also have, you can see the dimension and projection, so we can change the projection. If there was tiling, you could change that here. You can change the binning. This is a post binning, so this can basically reduce the size of your data set. So if I do a 16 by 16 bin, you'll see that my data set, my image gets much smaller. It's a little silly. This is really only something you need to use if you're doing a very, very large tiled image, and for whatever reason, it's not going to fit on the screen at once. So you can use that to reduce the resolution of the image. We also have this button here, which is poorly labeled. What this does is it allows you to assemble the image either by switching from a max intensity projection to slices if you have that data. If you don't have that data, it's not something you're going to change. I want to mention that there. We have the image viewer. So here's in second place, you can select the different well. You can see where you are. You can select the different fields. If you have different time, you can see the time. You can select the different Z's. So we have the first place up here to select the well. What data are you viewing specifically? Second place is down here. So again, we're going top to bottom, left to right. So now we've gone down here. So now we're going to go to the next panel, which is here. So we have the different views. If there was a 3D view, if this was 3D data, you could view that here. Oops. There's also thumbnail. So we've got the image view. In the image view, we can do side by side. So we can pick two different images from two different locations. So I could have well C8. I could pick a different field here. So you can pick multiple fields. We also have a quad viewer, so you can view up to 4 separate fields. This gets to be really nice whenever after you've done some segmentation and you have made measurements and you can look at the different wells. I'm always a big fan of, you put one up here, come on, then you come over here, you put one, maybe here. You could put one over at the corner. So this allows you to get kind of a better view of your data and see what's going on in different places. A thumbnail image allows us to load either every field, every Z, every time point. I like to load an image from every well. So this will load each well, and you can build up a nice plate map. There's a button up here in the corner, which allows you to save a snapshot. So we can go ahead and save a snapshot. And then if we look in the property of the reports, you'll see that there is a snapshot there. Come on, snapshot, open up. There we go. So it's not too exciting with this particular image because it's just a single row. But as you can see that you get kind of a little snapshot thumbnail of each image there. It's really useful when you're dealing with a big plate with a whole bunch of stuff in there. We also have cell information. Now the cell information, cell chart, stats chart, tracking chart, we're going to come back to that later after we've done some segmentation. But that's the basics of the viewing in CellPathfinder, okay? There's also the data. Go ahead.
I just wonder, is there a way to, before you take that snapshot, you can set a scale bar as well?
So in this snapshot, there's no scale bar that gets added unless you right click and set add scale bar. And then we put this on here. And then if you do a snapshot here and it says PNG JPEG, let's save it as JPEG and I hit save. And if I go to my reports and I open this report up,
cool.
Then it will get saved right there. So that saves exactly what you see. So you get a really ridiculous 49 micrometer scale bar because who would ever use a 49 micrometer scale bar? Can this style be changed? Why is it not a standard white bar? I don't know. It's not a standard white bar. You can change the color. So at least you can make the color white because it's standard for scale bars is white. You can turn off the show edge so you can get a kind of a white scale bar, but you can't make it like a thick, I feel like scale bars are always like a chunky white line and we can't do that. So, but what you can do is add it later. So, this is meant to be more like a digital caliper. So you get an idea of the size of some object. If you right click, that becomes available. You can select the pitch. The pitch means how much it's adjusted by. So if you set like 10 micrometers, it will now only adjust in 10 micrometer increments. If you select 1 micrometer, right click, select pitch 1 micrometer. Now you can get it to adjust in 1 micrometer increments. that will become very useful shortly in our next chapter, which is image analysis protocols. So let's go jump into that. Unless you have any questions. I mean, if you have any questions, please don't hesitate to stop, ask any questions, pass me a donut, whatever. It's all good. Yes, question.
Will we get access to this reporting?
Yeah, of course.
Okay, cool.
Typically what I do is, I know I'm flying through this. So if you want me to slow down, I'll slow down. If you want me to go faster, I'll go faster. Actually, going faster is a little bit hard, but you know. Okay, so the protocol. So with the protocol,
I
this
have
is.
one more question.
Please go ahead.
Can we access this software from our computers? Or is
No.
only-
It's only one,
Oh, sorry.
unless we
I was
buy it.
late.
Yeah, no worries.
Sorry.
Yeah, typically what folks will do is they'll install this on a central workstation and then use a remote connection and run it that way. You're still limited to one user at a time. Sorry, that's the way. I mean, that's an interesting question. Like, what's the economics and the philosophy behind the way scientific software is priced and limited? I mean, developers are expensive. The scientific imaging market is not huge. Companies want to make money, so they have to charge a lot of money to make it easy for people. And, you know, yeah, sorry, that's just the way it works. All the academics write their own, but it works for their data. And then, sometimes it works for other people. You know, a lot of people like ImageJ. ImageJ is the most flexible software. It has the most plugins. ImageJ can do everything. And as a result of doing everything, it makes it really hard to do some things, and training people how to use it is a real, you guys know, right?
Yeah.
I mean, I mean, back when I was in a lab, I wrote plugins for ImageJ. I think my lab plugin page is still up, so I understand, it's, if you're going to do something weird and write a one-off paper, it's fantastic. You can't beat ImageJ. But if you want to get down to business, and I have a plate with 96 wells, and each well has 25 fields, and I want to run the same analysis protocol and get a single CSV that has everything, good luck doing that in ImageJ. I mean, that's rough. But this to do, okay, I only have like, I don't know, 10, 12 wells here, but it's the same effort between 12 and 96 or 12 and 384. Doesn't matter. Okay, so how do we do this? So with the protocol, we start at the top, work our way down, go left to right. So we're just going to go through all of these steps. And that's it. So you typically start by loading. So whenever we load, it takes you by default to application. So you have to make a choice. If you have a pre-saved protocol, you can go over to user and load a pre-saved protocol. Nobody uses all users. We also have applications. So if you have a specific application, you can use one of these. A lot of times, most everybody starts out with a template. They're going to go and say, oh, I want to do dots in cell body. You click on that, and it tells you what the details. So it's going to go for time independent. It's going to tell you what the channels are. It's going to tell you what the object list is. It's going to have a brief description. It's going to show you what the linkages are and what the features are. If you were going to do something else, like let's say we just wanted to do nucleus, I'm going to start off with nucleus because it's easy. I'm feeling like I'm on a roll here, so I'm going to go with nucleus, and then we're going to come back and then do dots in cell body. Okay. So typically what you do is either load, or what I like to do is just use this button down here, to channel, which takes us to the next step. So I click on to channel. And so this takes us to the next step. So if you'll notice, we went from load, we skipped save, we skipped experiment, and went to channel here. Because if you just said the load, then you'd have to go back and step through all of those steps one at a time. If you use this next function, it basically takes you to the next step. And you'll notice that it's a drop down, and you can kind of skip ahead here. So in the assemble channel object, this is where we're saying, what are the channels that we're going to include for this analysis? You may add more or less channels to your heart's content. This gives you an opportunity if you want to do some mixed projections. So like I was working with a group where they were using the max intensity projection to do the analysis, but then they wanted to do, they actually had a 3D, kind of a 3D data set. It was a monolayer of cells, but they wanted to measure the total protein, and max intensity doesn't give that to you. So what they were doing is they had the max intensity, which made it really easy to do the segmentation, but then they also had a sum projection. And so they added a series of sum projections here, and they did the segmentation on the max intensity of the cells, and then they collected statistics on the sum projections to make that really easy. So this is a really flexible tool that allows you to basically build other channels, and we can use any of these channels for segmentation or measurement afterwards. Okay, we're going to go to the next step, which is to objects. So this is where we're going to tell the software what objects we're going to measure. I'm going to start out really easy, and then we're going to get complicated. So we're going to start out, we're going to measure some nuclei. We're going to tell it which channel. In this case, we're going to use channel 2. And then we can select a finder. Now, the finder is a preset protocol list. So it's basically the protocols are kind of like pre-built to make it easy for you to get started quickly. There's also an advanced. So the basic, it's a limited user interface. So with the limited user interface, it keeps you from making dumb choices, basically. Keeps you from screwing up. And it limits what you can do. The advanced lets you do anything. So you can get lost going down the rabbit hole using all the different steps to make a arbitrarily complex image analysis protocol. And do things. But we're going to start with basic, and we'll come back. We'll get to the advanced in the next round. I'm just going to start off easy. Now I'm going to go to algorithm. This is the next step where we go through and we set up our image analysis protocol. So in the nucleus here, now we've got our basic finder, and we have the sample of the cells here. So one of the things that you typically should do is you should actually open up multiple wells. I'm going to stick with 2 for the time being, just because this screen is relatively small for screen sharing. So I could set up, say, well C2, and then let's pick well C10, okay? And so what we're going to do is we're going to apply our image analysis protocol to 2 wells at the same time to make sure that it's basically a sanity check, to make sure our image analysis parameters are reasonable. So the first thing I like to do with the nucleus is to right-click and add a scale bar. And then I'm going to go in and find the size of these nuclei on the short axis. And right-click, use this pitch, 1 micrometer. And then I'm going to set that. Okay, so these cells are about 16, and I never trust just 1 cell because an N of 1 is a useless sample. And if you're doing, and that's why you guys are here. That's why you bought a CQ1, because you realize the N of 1 is a mistake. You need multiple measurements because you shouldn't be publishing anything with only just a few measurements, right? Right. Okay. Let's see here. Yeah. So that actually, I'm not doing a good job with that measurement here. Let me fix that. So these are about 15 to 20 micrometers roughly. Again, a perfect measurement is not required for this particular setting, but you can go in, get a rough idea of about 15 micrometers. Now this is important because we're going to go ahead and run the analysis. So I click on this little green button right here, and it's going to go through and run our segmentation and show you what it looks like. It's actually pretty good. However, there are a few that maybe aren't perfect. And so what we can now do is we can go into the tuning and try and improve that. So in the tuning here, so you see I'm clicking on this little button that says, this little up-down arrow that says tuning. We have a couple options. So this is the limited user interface for tweaking the image analysis protocol that I was talking about. So we can do things like increase the detection factor or change the nuclear diameter. So we could go in and change it to, say, 15 and see if that gives us a better segmentation. And you'll notice that when we changed that, we actually got over segmentation. So maybe 20 was a better choice. And now the other thing that we can do to improve the segmentation here, so that's pretty good, but we still have, this one looks good. These splits seem pretty good, but we have a little bit of over segmentation here and here. So let's look in the more, and we can actually go into the more section here and adjust the split factor. And if I decrease, if I 0.5, come on, 0.5. Numlock on. Okay, 0.5. There we go. And I run that. that should make it less likely to split and give us a better segmentation. So that those cells down here, that one's been correctly segmented, and it looks like most of these are pretty correct. I don't see, nothing is popping out. These are correctly split. Yeah, we're in good shape. So you can go in and adjust that. Now, if you do something ridiculous and you say, well, I want to see more cells, or you can come in and change these values. So if I put in a detection factor of say 5, it's going to come up, it's going to make it more sensitive, and it's going to come up with a lot of false positives. So you can see now we're getting all these false positives. Maybe there's some noise there. It's just being like overly sensitive. I typically run with like a value of 1 to 1.2, sometimes 1.3, and that gives me pretty good segmentation. There's a little something there, but I'm actually pretty happy with that. So real basic. Go ahead.
Like, what detect factor like actually changing in like the segmentation algorithm? Like, what does that correspond to?
In the basic image analysis, you're not supposed to know. That's the point of being basic, is it doesn't tell you that it hides. It's a black box. It's a light gray box instead of a black box. What it's doing is it's just there's some unitless value which is multiplying the number based on I don't even know, honestly. It's something with if we switch to the advanced, which I'll do later, I'll give you a little bit more visibility, but it's some multiplicative factor which brings up the background. It makes it more sensitive to the image. I honestly can't tell you exactly what the detect factor does. The higher the value, the more sensitive it is. If you increase it to the maximum, you get all sorts of garbage in there. Actually, this is a pretty clean data set, so there's not much. If you had a much more if you had a much noisier data set, you would get all sorts of garbage in there. But it's basically changing how it's an auto thresholding function that's being used, and it's changing how the auto thresholding behaves. So it uses size and this kind of arbitrary detection factor. There's actually other thresholding methods that you could use. So sigma starts with a particular, so this is T3 uses the sigma thresholding, and what that does is it looks at a particular intensity, and then it finds some sort of ratio between the adjacent pixels for objects. So it's more like a rolling hat type of thing. But T1 uses what we call the dynamic threshold, and yeah, I'm sorry, I can't give you a good answer on that one. I've actually asked the developers about that, and they say, it just changes the sensitivity. So, sorry. Yeah. Right, next step is to link. If you had multiple objects, you could link those here. And this is setting up a parent-child relationship between the different objects of segmentation. And this comes into play in the measurements later. The next step is to time lapse. If this was a live cell data set, we could do tracking. So we could do object region tracking to track new objects, motion prediction, search range tracking, daughter spells, and splitting, generations, and you could select multiple targets. So like, let's say you had cells that were moving around, you had vesicles, you could do that here. Time in same region is for like calcium dynamics or kinetics measurements, where you have objects that are staying in the same place but are having some intensity fluctuation. So we could use time in the same region to track that. So it doesn't redo the segmentation, it uses the same segmentation at all time points, and then just connects those objects in time. Time independent, it will treat each time point completely separately and rerun the analysis at each time point. Since there's only one time point, time independent works fine. Next step is to feature. Feature is where we specify the measurements that we're going to make on these segmented objects. So these are things like size, roundness, diameter, coordinate, or the position. Coordinate in well gives you the absolute position on the whole plate. Coordinate in well gives you the position relative to the center of each well. You can get things like intensity and the channel. There's also texture measurements. So if you want to get some sort of arbitrary value that gives you information about the texture of the intensity in the cell, you can select those. Then we run pre-analyze. Pre-analyze picks a subset of your wells to identify the objects. Since there's only like 12, there's only like what, 9 wells here. I'm just going to run everything because I know it's going to be really fast. But if you have like 3, 4 well plate, you would not necessarily want to pick all of them. You just want to pick 1 or 2 to get an idea. So this is going to go ahead and perform the calculation. It's going to take about 20 seconds on this, or maybe 30 seconds on this particular computer. Oh, I wish I had my other computer available. It's a little bit faster, like 4 or 5 times as fast. But anyway. So we've gone through and made the segmentation. And this is where we start to look at things like our cell chart or our stats chart. So now we could go into the stats chart, which is population statistics. I'm going to right click, select chart type, select heat map. And then in here, I can go in and do something like cell count. So this will tell me, I'm actually going to turn, sorry, hang on a second. I got to turn something off here. I actually skipped a step. So in the experiment, you can specify the type, the compound. So if you're doing like dose response curves, you can actually go back here to this experiment and you can set up the plate conditions. I'm sorry, I forgot that. Skip that. But I'm going to actually have to turn that off just to come back. So I'm going to select this to well. So you can actually right click and go into the settings. We can specify how this data is being processed. So well column times well row, or we could specify dose times series. I'm just going to do well column versus well row. So this is going to show you all of the wells, and it's going to show you the measurement that we select at the bottom. So in this case, this is the count of the number of cells in each well. We could do things like the average area. So if there was a difference in area on the different wells, we would get that here. We could also go down, look at, is the diameter different? Is the, let's see, let's go for the mean intensity in channel 2. So I could select cell nucleus mean intensity channel 2. And this will show you the mean intensity of the cells and show you if there's any differences. Now, sometimes you have to be a little bit careful because you'll see that the range in the values is from 3,000 to 4,200. That may or may not be, realistically different. So if I right click, go into view setting and go into series, I can see that I can change the heat map here. And we can also see the min and the max are auto selected. So these may or may not be appropriate values. So it might be that a reasonable min is 1,000 and a reasonable max is 10,000 to make reasonable comparisons between different data sets. And when I hit apply, we see that, in fact, the differences in intensity in these cells are not that much. So that might not be a reasonable, coloration there. So you just have to, be aware of things like that. So the stats chart gives you information about kind of well level information. If I go to the cell chart, this will give us per object, per cell information. So now I can right click, go to chart type, and select information about the scattergram, histogram, or pie chart. So let's say I do a histogram and I come in and I just want to do a histogram of the mean intensity of channel 2. So now this will graph a histogram for each of those wells for all of the cells in each of those objects, and you can see what those histograms look like. I can also right click, go to view setting, and we have some options here that are kind of fun. So we can actually collapse those. So this will put all of the wells into one. So now this is all of the wells graphed together. So then we can do some other fun things, like we could do the dosage or the series. So I could actually see with the different. Hey, I told you to change that. Why didn't you change that? Supposed to. There we go. So because I had the different dosage, I could actually go in and graph the different temperature on the same well. Or not the different temperature, the different measurements on the same well. So there's all sorts of things you can do here to adjust that.
Question.
Go ahead.
If you would want like specifically this data, but you kind of want to plot it, separately, can you like export exactly what you see here, like in terms of the raw data?
So once you've run the analysis, you go into Property Reports and click on this folder here, and all of your statistics are saved as CSV files here. Getting the data just for this this particular graph, that data you don't. I know it's kind of it's a little it's how to say it's a little bit of a bug. Maybe not a bug, it's a behavior. So when you say save CSV, that CSV file does get saved, and it is saved in here, but that data is in fact a duplicate. So you notice the size. This is that chart object that I just saved, but in fact it's a duplicate of the CSV file, which has all of the data in it. So if I were to open up an Excel, this computer has Excel on it. It actually has a spreadsheet of the entire all of the data. So it doesn't just put only this data. So yeah, I know it's a touch annoying, but what you can do is there is a snapshot function here, so you can click on that snapshot, save it, and then you'll notice that over in Property Reports that you get a snapshot of exactly that file. Come on. Again, it's not an editable graph. But, for something quick and dirty, it's okay. I mean, look, CellPathfinder is, it's a tool to get the work done quickly. It gives you the results. I would not say that it is a replacement for a proper statistical analysis software or for a graphing software. If you really want Excel to do your graphs, you should probably not use Excel. You should probably use something else. You should probably use like GraphPad Prism or, real software. It's, it's meant for that. CellPathfinder gives you a quick result. Like it tells you, is my data reasonable? Yes or no. And then you take that data out, you take those CSV, you throw it into GraphPad, you throw it into Gene Data, you throw it into whatever. And, something, what do you guys use? I mean, I saw some people grimaced at GraphPad. I mean, that's kind of the standard for the biologists typically.
The best one. We've been pushing to get a license for my entire PhD.
Are you kidding? Well, how much does GraphPad cost? Because I remember when I was in a lab, it was like $150 a year.
I bought my own. It's like $160 a year. Yeah, I did it.
Oh, yeah. That's chump change. It's nothing. That's like 2 pizzas or something, right? 4 pizzas.
For a
Easy.
PhD student, it's like a month of food.
You guys got to go on strike and demand better pay. That's actually going. No, seriously, that's going through the NIH. They passed a thing. They want the postdocs there's supposed to be a thing where the minimum average pay for postdocs is supposed to be $70,000 because they don't get paid much better than the PhD students right now. So, but that's changing. You're dying. Oh no. Well, yeah. Well, I mean, right now you guys get what, less than minimum wage, right?
Oh yeah.
Yeah, I should be working at In-N-Out and be making more.
Yeah.
Why should I be? I'm contributing human knowledge less than someone who's contributing fries to my tray.
Ouch.
Yeah.
Sounded personal.
I like In-N-Out.
Okay.
Okay, sorry guys, we're off track.
Yeah. Well, but don't
You were
worry. Once
triggered by
yeah, no, I understand. I've been there. But I mean, the bright side is that once you do graduate, and after if you do a postdoc or not a postdoc, then you get paid a lot more. So it still doesn't even out though. You should get paid better than minimum wage. But anyway, it's a whole lot. Sorry.
Thank you.
Yeah, I'm with you. So I'm supporting you. Yeah. Right. Yeah. So you should use GraphPad or something else like there's a like Statistica. There's a bunch of other graphing software like that you can use, or if you're into Python, you can do Matplotlib. Matplotlib. That's what it's called. It's kind of a pain. I mean, you can do it, but God, why would you?
Okay.
Okay, that's it in a nutshell. If you have more time, I'll keep going. We'll do something a little bit more complicated. So we're actually. Do you want to spend more time, or I forget how much time we have allocated here?
I'm just spend more time. What were you gonna say? Yeah, we're a few that want to like, but it's it's recording anyways. So.
I want to see the cytoplasm.
Yeah, it would be good to see cytoplasm segmentation maybe.
Like
Thank you
Okay.
so much. I
Yeah.
have to-
Yeah, Naomi is sneaking out.
Thank you.
Okay, thanks. We'll keep recording and we'll send this to you later. Okay, so I'm just gonna do this dots and cells. I'm gonna go back. We're gonna do the same basic thing here. All exactly the same. The difference here is that we're having additional objects. Okay. Now, one thing I got to change is this. For some reason, this particular data set's a little screwy. And the channels are swapped. Whatever. And then what I like to do here is I like to use an advanced protocol for Granule, because Granule doesn't work very well in this particular data set. So I'm going to go to algorithm. Oh my God, you've seen all this before. It's exactly the same. The difference is that up here is we have nucleus, cell body, and dots. Okay, that's the difference. Okay. So I'm just going to go ahead and tune this like we did before, and just have like a reduced split factor. 0.5. God, it just does it every time. There we go, 0.5. Comes out pretty reasonable. Go ahead.
That's just miscellaneous objects outside the nucleus and the cell body.
Yeah, you can call it whatever you want. You could call it puncta. You could call it vesicles. If you want to call it, what's your name?
Ethan.
Ethan's new vesicle of Ethan's new things. It's Ethan's new thing now, so you can call it whatever you want. So yeah, it's just an arbitrary label. You should give it something relevant so that when you come back like 6 months later, you're like, Ethan's new thing. Oh, well, that's Ethan's old thing now. What the heck are we talking about? Dots is just what somebody thought was reasonable. So, okay, so to do cytoplasm. So I just jumped right back into the cell body. This is one way to get segmentation. And if I go up and do contrast, I can kind of adjust the contrast. And you can see that this default cell body was actually pretty awful. So what I'd like to do is come in and do a label transform. And actually, before I do that, I want to just check. Okay, so what this did is it used the nucleus result, and then it used a method to kind of expand that out. It works okay. It's not the greatest. I mean, it's okay. So what I like to do is I like to start showing people about the kind of advanced functions by coming in here. I come to this next section, which is label transform. So these colored objects are labels. This is a segmented, we've gone in, we've looked at this data, we have segmented it, we've labeled it. So this is some contour which is going to define that object. Okay, so an arbitrary set of XY points which defines an object. So we're going to come to this label transform, and I'm going to select something because I happen to know, and this is why you did the training, is that we have a whole bunch of other things that we can do. We can transform the label into something else. So I'm going to use one called Expand Image 3D. And then in Expand Image 3D, it requires a second reference. So in this case, I'm going to use channel 1 as the source. So if I mouse over this, it'll show me expand image 3D. The effect is expand region 1 until image 1 in intensity stages. So what this does is it's effectively what's called a seeded watershed function. It's using the label in the input, so region 1, and then it's expanding it out based on the intensity of the image that we are specifying in image 1. So right here, this is region 1, so we can specify the output. So I'm going to use the cell body output, and then I'm going to use the image. So here, so I'm going to use channel 1 source, and then it's going to expand out until this intensity threshold that I input here, and then it's going to use a number of iterations to do a best fit. So I typically use like somewhere between 3 and 5 iterations to get a best fit of this intensity gradient. So it's also similar to like a level set method. It's not a level set. It's not a level set, and it's not a seeded watershed. It's not a seeded watershed. It's just a expansion based on intensity gradient. So if I mouse over here, I'm going to adjust my contrast a little bit. And I'm just going to kind of say what's background versus what's not background. So if I mouse over here, I'll see the intensity in the image up here. So as I move around, background is about 10, 19. If I look on the cells themselves, it looks like the intensity is about down to about 120. So 150 is actually reasonable. I'm just going to go with that. So now it's going to take those regions and expand them out to get a more accurate edge, because edges are very important, and that did not look like it worked very well. So I'm going to go and lower that value. So let's go down to like 110 maybe. Let's see if we get a little bit more. So that should expand those out considerably. So you can see now those edges are a little bit further out. So maybe let's really go lower. Let's drop this down to like 50. Okay, having a little bit of fun. So now it's going to expand those out till the intensity level is about 50. So now you can see we've got much more complicated edges, and hopefully it's a more accurate measure of the true shape of the cells. Okie doke? Cool. So now let's go find Ethan's new things. So this is now the advanced user interface. Basically, it lets you see all the guts and all the bits. And, you know, we could run this and see what it comes up with. So if I just go down to the very last, it'll come through and it'll show you what that looks like, and we can zoom in. You can see it actually does like kind of an okay job. Maybe it's missing a few, but it's actually finding a lot of puncta, and I can toggle on and off and come up here, close the little eye and toggle on and off those objects. I mean, you know, it's okay. It's not bad. I mean, we could just run with that. But I like to, if you guys have time, I like to go through and show you how I would approach this particular problem. So first I come in and I would get rid of all this stuff because I don't like it. And I'd get rid of this because I don't like it. You have to do the labeling. And so I would minimize, the first thing I come in and do is make the kind of the simplest, the minimum requirement for a segmentation is to have a binarization and a labeling step. You can't get away with anything less than that. So typically what I would do to find puncta is I would come in and I would add 2 Gaussian filters. I typically do what's called a difference of Gaussians. So I add 2 Gaussians and a subtraction, and then I add a threshold to 0 after that. So typically what I do is I kind of figure out how big these objects are, because scale bar, because it's all about the object size. And so I like to come in and say, well, how big are these objects? I got to right click, change my pitch to 1 micrometer. I say these are really tiny objects. So they're about 1 or 2 micrometers. They're only about a micrometer, right? They're puncta. They should be small. We'd expect them to be small. So I'll typically do about a 3 times the object size. and then usually about a 30 times the object size. And then what I like to do is to do an image subtraction. And so one thing you have to do here with this is that I don't see it's using a Gaussian on the previous output, and make sure this output is the channel 1 source. And then I have Ethan's new things Gaussian 1 minus Ethan's new things Gaussian 2. So I'm going to take the difference of those two, and then I like to threshold out to 0. So anything less than a value of 1, because one of the problems with the Gaussians is it does the operation in floating point, and you end up with like lots of negative numbers. So let me show you what the results of this are. So it comes in, it smooths the image, and actually we have a view here which is kind of neat, which is the before and after view. So here's the before, here's the after, and I can do the Gaussian and the other Gaussian. So this shows the image and it shows the smooth version. So what we're going to do is we're going to subtract this smooth version from the original image here in the subtraction. And this is going to leave us with the puncta as being much, much brighter. But I don't know if you can tell, look at the numbers up here. We've got a whole bunch of negative values. And image processing doesn't really play very nicely with negative values because negative intensities don't really make sense. So I like to run a threshold to 0 to remove all those. So now what I get is a much, much higher signal to noise ratio of those puncta. And it's going to be much, much easier to identify these puncta. These are very bright. I could use dynamic threshold. I typically don't like to use dynamic threshold. Typically switch to something else like threshold sigma. And I set threshold sigma. Now you'll notice that there's this red box and an arrow down here. And this means I can't do anything until I fix the red box. So I'm going to set that to previous output. So you've got to go ahead and do it. I'm going to turn off this step here. So now all I need to do is to go ahead and threshold these. I like to use threshold sigma for this. And I'm just going to go ahead and run that and see what we get. And that's a little bit too aggressive. So what I'm going to do is I'm going to set my minimum gray value to be about, probably about 200. And this should give us a pretty good segmentation for all these puncta. So that's actually pretty darn good. Now what I could do here, if I want to split these apart, or I could actually make this a little bit higher. I'm going to make this a little bit higher. I want to really just pick out the bright ones. Yeah, that's pretty good. That's really good. So now I have a couple choices. If I want to just count these larger clusters as one, I can do that. Or I can go in and get really fancy and I can do some binarized transforms. So one of the things I like to do with the binarized transform is I like to do what's called a maximum distance. So I can use find maximum distance. And what this find maximum distance does is it will take this previous, I don't want to do maximum, I take that back. I'm going to do find maximum image. And I'm going to actually set this, so this is going to take this binary and it's going to reduce and it's going to find the maximum intensity point in the center of each of these. So it's going to find the maximum point, and it's going to use that maximum based on the intensity of this previous image. Okay, so it's going to go through and find a whole bunch of individual points. So you see there? So we can do that, we can use that, and this is basically going to be a seed point. We can do a seeded watershed here if we want. And there's a couple ways we can do this. So I'm just showing you some, like, you know, how kind of crazy and in-depth that you can go here. And this allows us to come in and separate out structures like this. And then so in order to do that, then typically what you do is you do like a labeling here. So each of those are separately identified. And we're going to need to do an extra labeling. And this one, instead of using this find maximum, I'm going to use the threshold sigma up here. Okay, so I'm going to label this as an object, but I've also got these labeled as an object. And then in my next and last step, I'm going to use the label transform. And I'm going to use a different function, which is expand region. And I'm going to expand region 7 into region 8. And this will give me a segmentation. So now when I do this, it's going to take each of those and split that out into the threshold sigma level. So now we're going to get segmented objects So it's basically a seeded. That's how you break apart a seeded watershed into piecewise solution. So we're getting nice segmentations there, and we're getting these split apart. So that's roughly how I would do it. Now the next step would be go to linkage, and now we can connect the nucleus as the parent, and we have cell body, and then Ethan's new things are in here. So if we're happy with that, then we go to time lapse. Again, it's not a time series. We go to feature. Now, the one thing that here that's quite different is because there's a relationship, a one-to-many relationship of the puncta. I know you guys are getting tired. We're coming right in on that hour and 15 minutes, hour and a half. Your brains are like, I'm full. I don't want to hear this guy anymore. Shut up. It's Friday. Let's go home. We're almost done, guys. Almost done here. Okay. So the next thing that's really important is this linkage, which there's this one-to-many relationship between the puncta and the parent. So it's basically between the parent-child relationship. And so you have to specify those measurements. So if you want, like, say, the average, so we could get, like, by default, you always get the count. So we get the average count, but we can get the average area. We could get the coordinates, like the average coordinates. We could get the mean intensity of those puncta. We can get the average, the total, the median of the puncta. So we get one value per cell. But we're also going to get statistics about each individual puncta if you want that. Okay. I'm going to go to pre-analyze. I'm going to run this again. This is still a relatively fast analysis, so it's not going to take much time. Hang on a second. That should have been really quick. Yeah, okay. Preview. Is something screwy? No. Yeah, there's something screwy there. It should have taken longer than that. Cell bodies. Preview. Yeah, I'm not. Oh. Not sure why it's not showing the statistics. I mean, it should actually show that stuff here. Something funny is going on. Huh. Because normally then, once you're satisfied with that, you hit start, goes through and runs the analysis. And but I'm not seeing the objects here. So something screwy happened. Looks reasonable. Looks reasonable. We have to run this. That's reasonable. Oh, you know, I bet there's something funky. This is all good. I think this is gonna sound stupid, but I bet this apostrophe screws everything up. Because I don't think I've ever put an apostrophe in that before. Let me try that again. Apostrophe. Don't put apostrophes in the name. I'm sorry about that. Jeez Louise, that's
How does it work?
Take like a week to figure out.
Like, why is it working? You can access a file path because put a space.
Oh yeah, I know.
yeah. So clearly somebody didn't, for the programmers in the room, somebody didn't properly, escape the token and catch that unmatched apostrophe. Shame. Shameful.
How is it not a raw text string? Like, it should be, you can have an apostrophe, that should be fine. I think if you mix it will,
I don't use apostrophes. Apparently, I mean, I'm sorry. That's
it's still managed.
You use spaces for anything, I think.
I
I
was
use
using a
spaces.
string name.
Yeah.
And match those two string apostrophes together. And it's gonna shorten it and it doesn't know what it's looking for.
So is that like, so they get like an error.
Because not really, it can still run, like, you know, or it completes. Doesn't quite count as an error.
Oh.
okay. So what's supposed to happen is you end up with something like this. I like to turn on the object so you can come in and see what the cells are. You can see the cells. You can do fun stuff like filling them. And see what cells you found and what the nuclei look like. So if you want, there's also the opportunity to do gating. In the gating, you can come in here and we could gate these, which is basically to take your segmented objects and label them based on some feature. So we could label them based on the number of puncta or the number of Ethan's new no apostrophe things. So we could come in here and use the histogram, and we could pick something like, say, the Ethan's new things count. And we could say, we're going to have like maybe 2 classes or 3 classes. We could come in, make 3 classes here. We could say many things, few things, and no things. And then what you can do is you can actually adjust, you can edit these individually. So if we could have between 0 and 1, this would be, I don't know, what's a few? 1 and 3 is a few. And many things is 3 to, oh, I don't know, 50 is many, right? Oops, I clicked on the wrong one. Oopsie daisy. 1. So this is the greater than, and this is less than or equal to. I'm sorry, it's a strict greater than or equal to and less than. So that ensures that you don't ever have overlap. And this one is 3 to 188. There we go. So now the only catch with this labeling is that it is not applied until you hit the start button. And so so everything you've done up until now was a preview. It gets saved in memory. It's not actually written to disk. It's not saved anywhere. When you hit start, it actually saves it to disk. Now, because we ran the preview, everything was saved in memory. And in the start, I want to show this to you again. There's this fast analysis mode. It does not redo the analysis. All it does is it takes whatever's saved in memory and applies the labels or the gating to it, and then it saves it. So now we also have our gates. So we can actually turn on the gates, and we'll see what the different object classes are based off of the labels. So now you can come in here and do things like look at a chart type. We can turn on a pie chart, and we can get a count of the different gates here. So you can see that most of the cells have many things, many of Ethan's things. A few have few, and only in these 3 are there no things. So Ethan hasn't been to wells 1 through 3. He's definitely been to wells 5, 6, and 7, and he's been pretty busy over in 8, 9, and 10. So maybe there's something that's Ethan toxic in 2, 3, and 4. I don't know. A little bit of I don't know. So that's it in a nutshell. Whenever you run this sort of analysis, this is something else I like to point out to people. In this report, you will also get your stats and you will get different statistics. So you've got your few things. So this is whenever you see the cell, this is all of the statistics for each object. Whenever you see underscore stats, that is the per well. So it aggregates all of the statistics of all of the cells and all of the fields into a single value for a particular well. So if I open up cell stats, this will give you one row per well. So you'll see the row, the well, the row, the column. It'll give you the well name. It'll tell you the compound, the dosage. It'll tell you the control type. And it'll give you all of the statistics for a particular well. So population statistics. Okay. Now the difference between cell stats, which groups all of the cells, and these others, this is the cell stats, so it's the population per well, but only for the cells which are labeled few things. Okay. Now the one thing that I don't always forget to do is in the post-process section here, if you want to collate all of those statistics, you can turn on stats all and the dose stats all. This collates everything into a single file. So you'll have separate columns for each of these different statistics, but it will all be in a single file, because if you don't turn that on, each of these are saved as separate files. And then the final thing that we have to do, so after we've run this, is we need to not forget to save this with a reasonable name. So this is in cell body. So we can save that, and you can put any sort of reasonable description in here. And then that's it. And then in your report, you've got your data. Any snapshots that you've made will be saved here. Everything is here. Let's see, did I? Yeah, okay, save. So whenever you save the protocol, you can either save it as a user, you can save it all users, or save as. If you save as, it will save out a protocol which you can share with people. So we could save Ethan's things in the cell body. We can save that as a protocol. And then I could say, email that protocol to you, and then you could run that someplace else. Now, the next time, now ideally, the next day, somebody else comes in here, goes to protocol, they load, they go to user, they select Ethan's things in cell body, load, wait for a moment, and then they hit start, and it will just run that protocol with all of those settings. Everything is saved, and you walk away, you go get your cappuccino, you go get your Stanley cup, fill it up with the Starbucks iced latte, and you're good to go, right? Or, whatever. I mean, I'm just saying something like, I don't know, or, Arctic cup or Yeti cup. I mean, I don't know, or water. You should be drinking water, or, your soy latte macchiato, slushy. I don't know what people drink. I mean, the water. Yeah. So, and that's it, right?
Cool.
Cool. Yeah.
So I know that was a lot, but digest it. And, if you have questions, you should have, if there's any more questions. I mean, have you guys done any image analysis or image processing before? You've used ImageJ? Yeah.
Yeah.
Yeah. I mean,
CellPose. Lots of CellPose.
CellPose, yeah. Yeah, I mean, CellPose is really cool. It's easy as heck to use, but does it do this? No, I don't think so. Yeah. Yeah. Yeah, it doesn't give, yeah. Pretty cool. Yeah, it'd be nice if we could use CellPose to do the segmentation for us. That would make life easy and nice. One of these days we'll do that. Not today. So sorry.
that something that's integrated into the CellPathfinder software already? I thought it was only the in-house, like Yokogawa CellPathfinder segmentation algorithm.
Currently, that's the case. So CellPathfinder, I don't have it on this particular computer. We do offer a deep learning option where you can build your own deep learning model. It does use VGC16 if you know your deep learning models. It's VGC16 is the model in PyTorch if you like are into all that stuff.
Yeah.
I mean, it's not the newest, it's not the greatest. It works. Go ahead.
But like so everything we've seen today is only in the separate program CellPathfinder. It looks similar in the analysis you can do parallelized with your image capturing.
Yeah. So
is it the same or like just like in quick, what's the difference there?
So the CQ1 software does have image analysis built in. Basically, the big difference is that the underlying image is exactly the same. I'm sorry, the underlying engine is exactly the same. And in fact, you can export these protocols from CellPathfinder and load those protocols, with the exception of filament, like neurite tracing. Those protocols don't work. That's not available. And also the deep learning protocols are not available. Of course, you guys didn't buy the deep learning module, so it doesn't matter anyway. But you can actually export most of these modules from CellPathfinder, load them into the CQ1 and run them. The difference is that the CQ1 software has a limited user interface. So that basically you only get access to the basic options. You don't get access to the advanced functions. So yeah. Now it's interesting because even with the basic, you can do some image filtering and you can do label transform. So they do have some options, but they don't have all of these options available. You get a subset. So it's a subset. It's not the complete set. So most so there's two other things that are different. So kind of number one, CQ1 analysis is a subset of CellPathfinder. Number two, the CQ1 analysis software, you can actually set it up to image a well and then run a CQ1 analysis to get results immediately after imaging that well, and have it do that on a continuous basis. So that is an option with the CQ1 software, which is not available with CellPathfinder. Number two, there is a protocol or a special function called CVACE, or Cell Voyager Automated Cell Explorer. So we call it CVACE. And yes, ACE is named for Ace from One Piece, because apparently some of the guys who do programming are One Piece fans. That's really, they made up ACE, Automated Cell Explorer, afterwards. So what that is, CVACE is a separate program which controls the CQ1 software and CellPathfinder and links them together. And it is a multi-pass software. Basically, it goes in, CQ1 scans like a slide or a well to find something interesting, like say a spheroid, like my background here. So it'll go in and find spheroids, usually at low magnification. It takes that entire well, so it'll scan like the entire plate or the entire well. It moves that data over to CellPathfinder. CellPathfinder analyzes it, finds hits, so it'll find all the spheroids, and then it'll pass back a list of XY positions to the CVACE software, and the CVACE will then tell the CQ1 software to run a second analysis pass. So it might do a high resolution scan of each spheroid. So that's really cool, because let's say you have a low number of spheroids. Maybe you don't have like a large number of spheroids, or maybe you've got tissue and you want to just scan the tissue on a slide. You can use CVACE to identify that tissue and do the high resolution scan of just the tissue without having to image the entire plate. And you can do that automatically.
Cool.
So that is not available with the CQ1 analysis software. So that multi-pass acquisition is not available with the CQ1 analysis software. That requires CellPathfinder. So that's a difference. I'd say it's a pretty major difference.
Cool.
And also CVA. The other thing you can do is with CellPathfinder. So you can link it to a. You can actually, well, you can link both the CQ1 analysis and CellPathfinder to the CQ1 acquisition. So as part of the CQ1 acquisition, you can tell either one to run a protocol and analyze the entire data set afterwards. So that's the same, but.
Cool. Thank you so much.
Yeah, sure. So yeah, why don't you guys play with it? Send me a list of the names of the people, and I need their email. I don't need their names, just their email. I'll send you all the start pack information with all the quick start tutorials. Play with it, and then if you have more questions, we set up another session and go over this on specific data. Okay.
Sounds great.
All right. Thanks. Stop
Thank
sharing.
you.
Thank you,
And I'll
Arvin.
send you the recording later. All right. Take care.
Thank you. You too.
Thank you.
Bye.
Okay. Bye.
Thanks.
Whew, that was a lot.